Hey, today, ladies and gentlemen, we're talking about genetic engineering. Now, genetic engineering is a huge topic, so we're not going to get into a super amount of depth on it because basically scientists around the world spend their entire lives studying genetic engineering and basically altering genes. But I'm going to give you some of the basics of it, and your textbook will as well. So what is a genetic engineering? Genetic engineering is when you alter the DNA code of a living organism. So if you take the DNA in a living organism and you alter it somehow, we call it genetic engineering. There's different types of genetic engineering. There's selective breeding, recombinant DNA, something called PCR, gel electrophoresis, and transgenic organisms. Guys, there's also cloning, which you're going to read about in your book, and I'll talk about it a little bit here, although it's not in the PowerPoint. So what is selective breeding? Selective breeding is when you breed only those plants and animals that have desirable traits. So you find plants or animals that have traits that you desire, and then you breed those animals together, or you breed those plants together in order to get those traits. They do selective breeding a lot with dogs. Basically, you take a certain breed of dog that has traits that you like, and you make sure that you breed that dog with another dog that has those traits that you like, and that makes it more likely that the offspring will have those, again, traits that you like. People have been using selective breeding for thousands of years. We do it on farm crops, on domesticated animals. Basically, if you have a strain of corn that is resistant to disease, and you find that it doesn't get diseases often, you can genetically basically breed that corn with other corn of the same type, and you'll get more corn that is basically disease resistant. If you have an animal that tends to make more meat, or a type of cow that tends to have more meat on it, you would then breed that type of cow with other cows that have that more meat on them, and then basically you end up having baby cows with more meat, which you can then use. So we've been doing this for thousands of years with domesticated animals and farm crops. Again, examples are dogs. If you have people have a particular kind of dog that they like, they end up breeding that dog with another dog that's the same, so that they end up with the traits that they like. Also, people crossbreed dogs a lot. For instance, a Bishipoo is a cross between a Bichon Frise and a poodle. Okay, it makes this cute little fluffy dog. Okay. So if people decided when they did that breeding that they liked the cute little fluffy dogs, so they continue to breed them. We also do it with cows in order to ensure that we have disease-resistant cows and cows that make more meat and more milk. And again, people have been doing it with flowers and other crops, corn, all kinds of crops throughout for thousands of years. You might ask, wait a minute, Sullivan, how in the world did they do this 1,000 years ago if they didn't know about genetics or alleles or genes or DNA? Well, it was just observation. A farmer might observe that a particular type of corn was making a larger crop. He would then make sure that he planted that corn close to other types of corn of the same type, and that way he could get larger crops of corn. So basically, it was through observation before we learned about genetics. Recombinant DNA. What is recombinant DNA? Well, it's the ability to combine the DNA of one organism with the DNA of another organism. It started off in the 1970s with bacteria and has since moved forward. They've actually done it with fish. They've put fluorescent DNA, or DNA that allows the fish to be fluorescent, and inserted it into certain types of fish so that they can see them under the water for cleanup purposes. They've done this with multiple animals now as well, but it started with bacteria in the 1970s. So what happens, basically, you take a piece of DNA from one organism, and then you insert it into the DNA of another organism. So here you see you have a type of DNA here. You cut out the DNA that you don't want in the one organism, and you replace it with the DNA of another organism. It's called recombinant DNA. Why would we want to do this? Well, first of all, here's another example. You remove the bacterial DNA from the organism, then you cut it, and basically you insert the DNA of another organism in. Don't get too worried about all this detail. It's very complicated. All you need to know about recombinant DNA is you take DNA from one organism and you insert it into another organism. When you do that, the foreign DNA will actually end up showing up in the organism. Why, again, would we want to do this? Well, we're able to make actually human insulin for people who have diabetes using bacteria DNA. They insert bacterial DNA into an organism, and then that organism will make human insulin. Also, human growth hormone. Okay. If you have someone with dwarfism, they're able to actually produce human growth hormone using recombinant DNA and bacteria. They've also designed, inserted DNA into bacteria that will allow it to actually eat up oil spills. So when oil gets spilled out in the ocean, they've actually found DNA that they can insert into bacteria that then will allow that bacteria to be able to eat the oil, and therefore clean up the ocean or clean up the shores. So another way to do genetic engineering is to basically alter the DNA of plants and animals. In plants, that we do this a lot. We do it in order to produce disease-resistant and insect-resistant crops. We do it to produce hardier fruit. 70 to actually 75% of the food in the supermarket that you eat is genetically modified. If you ever heard of GMO food, it's called genetically modified food or genetically modified organisms. And basically 70 to 75% of all the fruits and vegetables in the food you eat in the supermarket has been genetically modified to either be a disease-resistant crop or an insect-resistant crop or a hardier fruit that'll last through cold spells or better or longer. So you're eating a lot of genetically modified foods. On the plus side, this is a good thing because it allows us to produce more food, which then allows more people to be able to eat. On the downside, genetically modifying our plants could possibly have adverse effects to people who are eating them. For instance, if you were not allergic to a regular tomato, but they genetically modified that tomato, now all of a sudden you might be allergic to it, even though you weren't allergic before. This is a lot of the ways that when people, there are people who've eaten a certain food their entire life or for a long period of time, and then all of a sudden they eat it again and they get an allergic reaction. Many times they believe that's because they happen to eat a genetically modified version of that food, which then caused a reaction because the genes are different. Corn, they genetically modify a lot. So again, one of the risks is that you can actually develop an allergy or you can be allergic to the genetically modified version of a particular plant, even though you're not allergic to the regular non-modified version. Again, they do this a lot with crops. So how do you create a genetically modified plant? Don't get too hyped up on this. It's okay if you don't understand all of it. Again, it's a pretty technical concept. But basically, they take bacteria and they insert DNA into that bacteria. Let's say you have a plant that gets sick a lot. It gets diseases a lot. They'll take DNA that will make that plant disease resistant and they'll insert it into bacteria. They will then insert that bacteria into the plant, and when the plant breeds, it then ends up being disease resistant. So they'll take DNA from basically DNA and put it into bacteria with the genes that they want. Then they allow that bacteria to infect the plants. And then that desired gene gets into the chromosomes of the plant and ends up being bred with the plant when the plant breeds. And then you have that new DNA and new plants, which makes that plant disease resistant or insect resistant. What do you think about eating genetically modified foods? We're going to do a Socratic seminar about this over the next couple days, but the question really becomes, should we be genetically modifying food? What kind of health issues might that have for us? Again, you could have allergies to the genetically modified food that you don't normally have. Our DNA inside of us, was it going to affect us at all when we genetically modify food? So that's something we're going to debate over the next couple days. Genetically modified organisms are called transgenic organisms. So if an organism has been genetically modified, we call it transgenic. Some examples of transgenic animals. Mice have been used and genetically altered in order to be able to study the human immune system. They've taken mice and genetically altered them to make their immune system more like the human immune system so they can study them. Chickens have been genetically modified to be more resistant to infections. Cows have been genetically modified to have more milk supply or increase their milk supply and have leaner meat. Goats and sheep and pigs have all been genetically modified to produce human proteins in their milk, which then scientists can pull out of the milk and use in people. So we do a lot of genetic modifications nowadays. For instance, let's say you need a particular protein to make a medicine for someone. What you would do is you would insert the human DNA into a goat cell, into basically a goat zygote, and then when that goat reproduces, or when it would basically have that human DNA within it to make that protein. That protein would then come out in the goat milk. So this goat actually contains a human gene that codes for a blood clotting agent. That blood clotting agent then is pulled out of the goat's milk and used to insert into humans who have a difficult time clotting their blood or who have some sort of disease where their blood doesn't clot well. So again, how do you create a transgenic animal? Well, basically you insert the DNA that you want from a human into the egg cell of that animal. And then when that animal reproduces, the baby animal has that DNA within it. That's just a little joke there. Congratulations, you're the first victim of human recombinant DNA. Basically, they took some sort of animal DNA and inserted it into him, and now he's all hairy. They also use genetic engineering for crime scenes a lot. Polymerase chain reaction, or PCR, basically allows scientists to make copies of a piece of DNA. So if they go to a crime scene and they find a little itty bitty bit of DNA, they will use PCR to actually make copies of that DNA so they can run tests on the DNA to see who it belongs to. Don't worry about this too much, guys. This kind of tells you how they do it. But basically what they're doing is they're taking a little piece of DNA they found at the crime scene, and then they're making copies of that DNA so they have more of it, so they can use it to do genetic testing to see who is the actual person that committed the crime. They also use gel electrophoresis on DNA at crime scenes. What they do is they insert the DNA that they find, or the enzymes that they find, into a gel, and then that gel makes these bands, and you've probably seen these before in different pictures. The gel makes these bands, and then each person's DNA has a unique set of basically lines or bands that show up, and they can use this to identify whoever did the crime. So they'll take a little bit of the DNA, the enzymes, and they'll put it in this gel, and then it breaks out into these bands, and based on the bands, they can then tell who committed the crime. That's about it for genetic engineering, guys. Here's some other links if you want to kind of go look around at it.